Hi everyone, I'm Erin Molloy, and today I'll be presenting work with Arun Durvasula and Sriram Sankararaman on using maximum likelihood network orientation to estimate admixture graphs. So it is increasingly recognized that admixture, which is the exchange of genetic material between populations that were previously genetically isolated, is a really important and pervasive force in evolution. A well-known example is gene flow from archaic humans, such as Neanderthals, into modern human populations. Of course, we don't directly observe admixture or gene flow, but instead we seek to infer these events from genetic variation data. Specifically, we are focusing on admixture graphs that model changes in allele frequencies as a neutral Wright-Fisher process with mutations occurring under an infinite sites model. A mutation immediately results in two alleles, shown as red and gray dots, at a locus, and each generation of alleles, shown as a row of dots, is created by randomly sampling the alleles in the previous generation. This random process can lead to changes in allele frequencies, referred to as genetic drift. So we want to look at the shared genetic drift between populations to estimate the admixture graph. This model is parameterized by a network topology N and a set theta of numerical parameters, including branch lengths and admixture proportions. So if you look at the network topology on your right, you'll see some vertices within degree 2 representing admixed populations. If there is no admixture, then the network topology is actually just a tree. And you might recall that tree space grows exponentially in the number of populations at the leaves. Network space is far larger than tree space, and this is a significant challenge when it comes to admixture graph estimation. In addition, likelihood functions can be computationally intensive to evaluate. In the cases we are considering in this talk, these compute-intensive likelihood functions can be avoided using summary statistics. F2 statistics, for example, measure the shared genetic drift between two populations. So given an admixture graph like the one shown on the left, we can compute the expected value of an F statistic for a given pair of populations. This statistic is related to the paths between the two populations. For example, the paths between populations A and C are highlighted in orange and blue. Now, on the right side, the observed F2 statistics are computed from genetic variation data for all pairs of populations. Methods that use summary statistics typically look at the residuals, that is, the difference between the observed and expected values, to evaluate model fit and compute an approximate likelihood function. Because we are using this faster approximate likelihood function, our main concern is the exploration of network space. So again, exhaustively evaluating all possible networks is computationally intractable for large or complex graphs. And in some cases, researchers will have prior knowledge which will enable them to manually construct a subset of networks or hypotheses that seem promising to evaluate. Alternatively, search heuristics have been developed in an effort to automate this search process while being computationally efficient. For example, in a likelihood-based framework, we could apply some operation to change the current network topology. Then we would fit the numerical parameters to the updated topology and search from the resulting network only if it has a higher likelihood than the initial network. This type of strictly hill-climbing approach is taken by the popular method TreeMix, which was developed by Joseph Pickrell and Jonathan Pritchard. So our main goals are to have methods that are not only completely automated and computationally efficient, but that are also accurate. Specifically, they recover the correct network topology. In this talk, we are interested in the case where search heuristics like those used by TreeMix fail to recover the correct network topology, as well as how we can improve the search heuristics in such cases. So our work begins with a case study, and we're considering the model admixture graph shown here, which has branch lengths and coalescent units multiplied by 1,000. And this model implies a vector of F2 statistics, which we're denoting X star. Now we consider how TreeMix, or a related method, would perform when given X star as input, and this is representing the case of infinite data because our estimate of the F2 statistics from genetic data would converge to X star as the amount of genetic data goes to infinity. So the first step at tree mix is to compute a starting tree on all populations via maximum likelihood. And because there are only 5 populations, we can compute the likelihood score for all 15 unrooted trees on the 5 populations. I'm showing the tree with the highest likelihood score, denoted N0 in the center, and it's rooted at the outgroup E, even though the likelihood function does not allow the position of the root to be identifiable. And for our case study, the ML tree is actually the same as the tree computed by running neighbor joining given X star. In the next phase of the tree mix method, an edge representing admixture or gene flow is added to the tree or starting tree N0. An edge addition is going to take an ordered pair of edges. It will subdivide each of the edges with a new vertex and then add an edge between the 2 new vertices, and that's representing gene flow. Now, the optimal graph from performing an edge addition to N0 is shown in the center, and it's easy to see that the resulting graph differs from the case study graph because the case study graph N star has A as the admixed population rather than E. And notably, the starting tree N0 cannot be transformed into N star with an edge addition because the starting tree is not one of the base trees of N star. So there are 2 base trees for N star. One has A and B as siblings if we go up the orange admixture edge, and the other has A and D as siblings if we go up the blue admixture edge. Okay, and tree mix performs other operations to search for a network of higher likelihood, namely it performs tail moves. And this begs the question whether we can traverse from our initial graph, which we just found, or we'll call it the starting graph, to our true graph N star, which we're calling the ending graph on the right. And we want to do this traversal using tail moves. So I'm showing a tail move here. It relocates the source of the edge in orange. So it moves that source vertex from the edge incident to C to the edge incident to B. And with a brute force analysis, we found that it requires at least 3 tail moves to transform our starting graph into the ending graph. Note that the graphs are different in terms of the position of the root, but again, this doesn't impact the likelihood score because the position of the root's not identifiable. Other than that, it must be ancestral to the admixed populations. So we could just reroute the graph. Now, this is one such sequence of 3 tail moves. It's important to notice that for this sequence, the likelihood score of the graph after doing tail move 1 is worse than that of the starting graph. So in a strictly hill climbing approach, we would not choose to do that tail move. But we also evaluate the likelihood of all networks with one admixture event on these 5 populations, and we found that our starting graph had the second highest likelihood of all networks in this space. It was second only to the ending graph, which is our true network N star. And as we can see from the residuals, the N star network is a much better fit for the data than the starting graph. And so in this case study example, tree mix gets stuck in a local optimum and returns the incorrect graph. And so we want to escape this local optimum, and to do so, we are interested in applying a single operation that would allow us to move from the starting graph to the ending graph. Because again, even if we were allowing tail moves to traverse through graphs of lower likelihood, the space basically of doing 3 tail moves makes it unlikely that we would get to the ending graph. So we want to think about whether we could get from the starting graph to the ending graph in an easier way. And we can do this actually through reorientation. So we will suppress the root and undirect the edges of the starting graph, and then reorient the edges of the undirected graph by specifying A to be the admixed population, that vertex incident to the edge incident to A, And then we will also specify the position of the root so that E is the outgroup. And we don't know necessarily which population to specify as admix, so we would need to try all of the purple vertices, but this is a lot fewer graphs to look at than doing all of the tail moves. And so the idea would be we could incorporate a maximum likelihood network orientation as part of TreeMix's search heuristic. To implement this idea, we utilized recent results from Huber et al. They proved that the orientation of an undirected network is uniquely defined by specifying which vertices or populations are admixed, as well as the position of the root. For an undirected network with one admixture event, then, the number of possible orientations has an upper bound that is linear in the number of vertices if we fix the position of the root at an outgroup. And so this orientation space is a subspace of the networks with one admixture event. And now we can define the maximum likelihood network orientation problem. So given an undirected network, we seek an orientation that maximizes the likelihood function. And we conjecture that this problem is NP-hard when the number of admixture events is unbounded. But when the number of admixture events is relatively small, we can exhaustively search orientation space. And we've implemented an exhaustive search for the MLNO into TreeMix's search heuristic. This software package called OrientaGraph is built on top of TreeMix, and it enables some other things. For example, the user can provide a network from which OrientaGraph will search for the maximum likelihood network orientation. And this will additionally show if multiple orientations have similar likelihood scores. Okay, so to benchmark OrientaGraph, we searched recent literature for model admixture graphs that could be used in addition to our case study. The most complex graph here had 10 populations and 2 admixture events. And we started by evaluating OrientaGraph given the F2 statistics implied by these models, which represents that case of infinite data. So while TreeMix recovered the correct topology on 3 out of the 8 models, OrientaGraph recovered the correct topology on 7 of the 8 models, so improving the performance of TreeMix in 4 cases. 3 of these cases are because of maximum likelihood network orientation, and 1 of these cases was due to the fact that we also extended the number of edge additions explored by TreeMix when implementing OrientaGraph. We also evaluated a new method called MicroGraph, which is also based on F statistics, but it operates quite differently compared to TreeMix and OrientaGraph. It recovered the correct topology on 5 of the 8 models. However, we did limit the running time of MicroGraph to 3 hours. In contrast, TreeMix and OrientaGraph always completed in less than 30 seconds. And on these modest problem sizes, it was fast to incorporate in an exhaustive search for the maximum likelihood network orientation. It's also interesting to note that MicroGraph could not recover the correct topology for the 2 models that are starred because of the topological constraints that MicroGraph imposes. And we observed the same trends in a simulation study using genome scale data. So here are the results. On the top plot, I'm showing the log likelihood score of the estimated admixture graph. And on the bottom plot, I'm showing the topological error measured as the triplet distance between the true and estimated admixture graphs. So lower error is better. And we are looking at how TreeMix in black and OrientaGraph in gray perform giving increasing numbers of loci, or increasing data. In all cases, giving a sufficient number of loci, OrientaGraph returns the true graph, so error is 0, whereas TreeMix does not. And importantly, there is a difference in the likelihood scores, so the graphs returned by the 2 different methods are not equally optimal. So to summarize, our case study showed that even simple models can be challenging, so that methods based on heuristic search get stuck in a local optimum and return the incorrect admixture graph topology. In our study, maximum likelihood network orientation improved method performance and was fast for our modest problem sizes. However, there was that one model shown on the right for which OrientaGraph returned the incorrect topology, and we want to investigate this in future work and try to improve OrientaGraph in this scenario and also looking into more complex admixture scenarios with more populations. With that, I'd like to acknowledge my co-authors again, as well as our funding from the NIH. A big thank you to the members of Shriram Lab for their feedback. And also, I developed Orientograph on top of TreeMix, which is open source and very nicely written. So I'm very grateful to the authors of TreeMix, Joseph Pickrell and Jonathan Pritchard. And lastly, thanks to all of you for your attention.